So some things to take in consideration is that the gene sets, they overlap. So in many cases, many of those pathways that you see, so gene ontology, the hallmarks, the transcription factors, reactome, and KEGG, they will have similar information. And within those databases, there are many terms that are similar to each other. So I would always recommend to run different couple of ones and take the overall consensus information that you get out of it. So some considerations. There is still some biases in gene sets because these are curated by the knowledge that we have. In the last year, one bias that we get is COVID-associated data sets, even though many of the COVID pathways are actually the same thing as any viral infection. If you put the gene list, then you'll find like gene terms that are enriched for COVID, even though I don't have COVID in my data set, just because those genes are exactly the same as any other viral infection. So there are gene set names that can also be misleading. So depending on the version of the pathways, some of the gene names also have changed. So in some cases, it's always good also not only to check the version of the data set that you're using, but also check the genes that you have in your data set. Consider also the gene set size. So usually you would filter. So for example, immune response is a quite big term or concept. So there are thousands of genes in there. So that might not be very significant, very important for you. So you can actually filter that term and not consider terms that has so many broad terms. Especially for single cell, the broader terms are actually not useful. So yeah, usually you would try to filter positive or negative expression of those genes and run them independently. Yeah, and always try to revisit your gene level data to see if they are actually significant. So for example, if you find viral response, then go to the gene list and see which genes are the ones that are enriched and plot them. And then you can see in the different conditions that you're comparing that all those genes are also upregulated or clearly upregulated.